Okay, well, thank you very much for the introduction. And I'm very grateful to the conference organizers, and especially Neil Darks, because I requested this slot just 3 weeks ago, this clinical trial just very recently, and so we consider it a late breaker of sorts. Most talks in this conference, of course, have discussed vaccines that stimulate antibodies. We've also heard talks about stimulating different kinds of T cells, including earlier in the session. These can be cytotoxic T cells or otherwise. What I'm going to be talking about is a newer concept, which is stimulating regulatory T cells, which can act to selectively immunosuppress inflammatory and autoimmune disease. Key consideration is our GI tract. Now, we know that we don't make immune responses to food or gut flora. And data has come out that shows that immune responses in the GI tract are generally controlled by induced regulatory T cells. And these cells play important roles in inflammatory diseases, autoimmune, and malignant disorders. So it's an area of very active investigation. So that's one part of the equation. Now, another part of the equation is an antibody which is directed against the CD3 molecule on the T cell surface. This is actually the first licensed monoclonal antibody, licensed 25 years ago, used for reversing renal allograft rejection. It's a very potent antibody. In fact, it's so potent that it induces strong mitogenic effects and binds the T cell receptor and can induce cytokine storm. So it has the right properties of immune suppression, but the properties of the antibody are much too acute. There have been more recent investigations in trying to downplay the pathogenic effects of this antibody, and these have had some mixed success in the clinic. But it looks like the specificity is right, and the oral route offers certain promises. So about 8 years ago, Howard Weiner at Harvard Medical put these two parts of the equation together and hypothesized that oral CD3 monoclonal antibody could bind to T cells in the gut-associated lymphoid tissue. This would stimulate regulatory T cells, which could suppress pathogenic T cells in autoimmunity and inflammation, a mechanism that's very distinct from injected anti-CD3 antibody. Another way of viewing this antibody is ingested orally. It makes its way through the stomach and intestine. It's presented to the gall, where it's taken up into mesenteric lymph nodes. Regulatory T cells are induced without the antibody becoming systemically available, and the regulatory T cells can then circulate in the body to various target organs, which include pancreas for type 1 diabetes, adipose tissue for fatty liver disease, brain for multiple sclerosis. So that was the hypothesis, and this hypothesis has been borne out repeatedly in experimental mouse studies. This slide shows a broad range of inflammatory diseases, which are the first three, which include diabetes, hepatitis, inflammatory bowel diseases, as well as autoimmune diseases and other inflammatory diseases in which anti-CD3 antibodies have been shown to be active. Preclinical studies then have shown a broad range of effectiveness in a wide variety of models. A safety profile very distinct from the injected antibody. Again, the injected antibody can induce massive cytokine release and cytokine storm and generalized immunosuppression, whereas oral administration does not. And in fact, oral administration in mice does not affect the level of CD3 in circulation in mice, again showing that T regulatory cells are induced, but without suppressing CD3 cells CD3 cells systemically. Studies have shown that the antibody is taken up within about 30 minutes to an hour in the intestine and can be found in mesenteric lymph nodes. The Tregs can then be shown systemically available within 1 day. They can be shown at sites of disease, and as shown in the previous slide, active in various efficacy models. Furthermore, these regulatory T cells can mediate adoptive transfer of suppression. And some idea about the phenotype of this suppression is understood because antibodies against TGF-beta or LAP being an important cell surface molecule in regulatory T cells, will abrogate the suppression that is mediated by these regulatory T cells. So it's a TGF-beta dependent phenomenon, and LAP is an important signature on these Tregs. So I'm now going to get into clinical data. First, a brief summary of a safety study that was done 2 years ago to show safety of this approach. This was done in 18 subjects, 15 of whom received the antibody in 3 different dose levels. Some also received an oral adjuvant. They were monitored then for safety and immunological effects. There were no treatment-related adverse effects. Safety and tolerability was excellent. There were no changes in CD3 blood counts. Blood chemistry and blood cell counts were all totally normal. There was a clear induction of regulatory T cell markers and anti-inflammatory markers. So this was consistent with the data in mice, showing very good safety and showing immune modulation. And that then laid the groundwork for proceeding to a phase 2A study, which is what I'm going to report now. So this study looks at NASH, non-alcoholic steatohepatitis, also known as fatty liver disease, and also enrolled subjects. These subjects have altered glucose metabolism, and many of them also have type 2 diabetes. They have a variety of conditions that lead to significant morbidity. Fatty liver disease has not been so well recognized until recently, but it's now been shown to affect millions and tens of millions have milder forms of disease. The incidence of disease rises with the degree to which an individual is overweight and is characterized by fatty cell infiltrates that cause acute inflammation in the liver and leave subjects at high risk for cirrhosis or liver cancer. So the study design was 4 groups of 9 per group. It was randomized. All the subjects were blinded. There were 3 different dosage levels and a placebo group. Again, we picked 9 per group as a good number for safety and to show certain effects. It certainly was not powered for any statistically significant results. We were only looking for trends. The primary endpoints were safety and immune modulation, that is induction of regulatory T cell phenotypes. Secondary endpoints were to see some indication or trend in efficacy of various efficacy biomarkers. Safety was monitored at the time of enrollment on days 7, 14, 21, and 30 during a 30-day dosing period, and 30 days after the end of dosing on day 60. Immunology and efficacy markers were followed pre, midway, and at the end of dosing, and 30 days at the end of dosing. The study was conducted at Hadassah Medical Center in Jerusalem. Safety, there were no treatment-related AEs. Safety and tolerability, totally indistinguishable between any of the dosing groups and the placebo group. No changes in CD3 lymphocyte counts, CD4s, CD8s were unchanged. All blood chemistry, electrolytes, and blood cell counts were completely the same when comparing all the dosing groups to the placebo group. Looking immunologically, there were increases in regulatory T cell markers consistent with inducing Tregs, and I'm going to show some of that data for CD4, CD25 lap plus T regulatory cells, as well as TGF-beta, which is kind of a major mediator of inducing regulatory T cells. In addition to this, there was indication of antibodies against this anti-CD3. The anti-CD3 is licensed OKT3, which is a murine antibody, and we tested the HAMA response, which is the human anti-mouse IG response, and did find that some of the subjects responded. This is consistent with the ability of the antibody to enter the lymph nodes and be recognized immunologically, but not seeing depression in CD3 means the antibody was not available systemically. The very surprising results in these small cohorts was we saw changes in efficacy biomarkers. They were positive statistical trends, and some of them actually were statistically significant. These were in AST, it's a liver enzyme, so reductions are consistent with reduced liver inflammation, reduced glucose levels and reduced insulin levels, which are favorable for subjects with type 2 diabetes, which included many of the subjects in these cohorts. I'm now going to rapidly go through some of the clinical data just to give you a little flavor of some of the effects we saw in these small cohorts. Shown here is the changes in CD4, CD25 lap positive regulatory T cells. This was a pool of the 3 treatment groups, 27 individuals compared to placebo. They were normalized to 100 at day 0, and you can see changes, increases in Tregs, which are all higher than those in the placebo group, and at day 30 we showed a statistically significant increase of Tregs compared to the level in the placebo group. Here is TGF-beta, again normalized to 100 at day 0. We can see significant trends, which were not significant, but still trends at day 30 and at day 60, which is 30 days post the dosing period. Higher levels of TGF-beta in all of the 27 treated individuals compared to placebo. Shown here is AST, liver enzyme, and you can see across these data not being normalized, but day 0 is almost exactly the same between the two groups. You can see there were reductions in AST levels in the treatment groups compared to placebo. There were statistical trends at days 21 and day 30. Shown here is glucose, and again, lower levels of glucose in the treatment groups compared to placebo. Likewise, insulin, reductions at day 30 and at day 60 with a statistical trend at day 14. So to summarize, data in phase 1 and phase 2A clinical studies have shown that oral anti-CD3, safe, well tolerated, no treatment-related adverse effects, favorable immunological changes in terms of inducing TGF-beta and regulatory T cells. And the phase 2A data show favorable changes in efficacy biomarkers, including liver inflammation, glucose, and insulin. Based on this, it's possible that oral anti-CD3, when used to induce regulatory T cells, could be useful in a range of inflammatory and autoimmune diseases. Obviously, each will have to be studied on its own merits. And our future plans are to initiate additional phase 2A studies, to develop a new monoclonal antibody, and also to proceed to a phase 2B clinical study in a leading indication. And in the interest of time, I'll stop there and be happy to take any questions you have.
We have a mic back here for this gentleman.
So one explanation for the findings could be if the patients receiving the anti-CD3 had weight loss, because again that would reduce insulin and potentially reduce the liver enzymes. Do you have data on the weight comparing the placebo group with the active treatment group?
So I'm not sure I totally understand your question. You're saying what is the explanation for the changes we're seeing?
No, so the changes could easily be explained by weight loss. So if you had greater weight loss in the group receiving the anti-CD3, that could explain the reduction in insulin and blood sugar that you observed. So were there any weight differences in the two groups?
From the overall randomization, the subject demographics were similar from one group to another. So I can't say there weren't any marked differences in any of the particular cohorts in terms of characteristics like that.
No, I mean over the course of the trial, did the people with the anti-CD3 treatment lose weight from the start of the trial to the end of the trial?
No, there were not significant weight losses across the study.
Okay.
I have another question here.
Did you see any patients with hypoglycemia? Hypoglycemia?
Can you ask without the microphone?
Oh, sorry. Did you see any patient with hypoglycemia?
Hypoglycemia. No, we did not. From a safety point of view, the tolerability was good. None of the glucose levels was dropped to any alarming level. Most of the changes in glucose were reductions on the order of 5, 10, 15%, so they were not, didn't cause any clinical alarm.